hello, hello. Can you hear me?
Yeah.
Great, thanks.
So I think we can all switch on our videos, or Alexandra, you are first. So
Yep,
you
sure.
actually share your screen, I guess.
I will do.
Also, so we need to prepare the slide summary. Is it ourselves?
Yeah.
Okay. I'll find it.
But we will go by order, Anthony. So you will be second. So
Okay, I will try and find it. I will get it.
So I'm just going to share my screen with you guys. Hopefully everyone can see my, oops, that's the wrong one. Hopefully everyone can see my slide.
Yeah.
Yeah, great. So my name's Ali. I'm a biomolecular archaeologist, so I'm very much coming at the role of the brain in metabolism from a very different standpoint. And it's been really exciting to hear about all of your clinical research. So what I'd hoped to be talking with you about today was the results of a combined proteomic and lipidomic investigation that we're conducting on preserved central nervous system tissue, which is far more commonly preserved in the archaeological record than most people imagine, even within our own field of archaeology. Unfortunately, due to COVID, the project has only just begun this week, but on my screen at the moment, there's some preliminary results of some preliminary investigations. But what we're excited to be looking at is 5 preserved brains of 13th century Franciscan monks from a site in northern Denmark, and also the preserved spinal cord of a 15th century pilgrim from York in the UK. We know from documentary historical sources that the Franciscan monks had an extremely calorie deficient diet. They lived in conditions of near starvation and poverty, whereas our pilgrim was a very wealthy man with a calorie rich diet. We also know from his skeleton that he suffered from DISH, which is clinically associated with obesity. But at the moment, we have very limited quantitative methods for studying things like metabolic disease and nutritional status in the past. So my question for you guys is, if we're able to recover ancient proteins and perhaps also lipids as well, what can we learn from them? Can we look at elevated levels of certain peptides as metabolic signals of nutritional deficiency, or conversely, accumulation of polyunsaturated fatty acids as signals of metabolic excess? So we're really excited to explore the bioarchaeological significance of ancient brains, and I'm really excited to hear your thoughts and answer any questions that you might have about my research. Thanks for listening.
Okay, thank you very much.
Thank you.
Open for questions. I know this is a little bit awkward with not seeing the poster. Let me start off while everybody grabs their thought. I had a problem with your poster that I didn't quite understand the amino acid composition.
Yeah.
That you showed. This is the amino acid composition of a protein?
This is the amino acid composition of all of the proteins that we were able to recover from our ancient brain samples. So what we're finding is it seems to be preferential preservation of transmembrane proteins. So leucine, for example, which appears to be the most abundant amino acid, is obviously highly abundant in the alpha helical regions of transmembrane proteins. So that's what we imagine is contributing to the overall amino acid composition in that respect.
Yeah, so I understand that then, but I then don't understand is you seem to conclude on your poster that there was an enrichment of some amino acids compared to modern man in the same sort of, and I can't really follow that, because if you have a codon that defines an amino acid at a certain position, it seems to be very unlikely that evolution has worked on that sort of time scale to change the codon usage to make that many differences in the same proteins.
Yeah, you're absolutely right, Frank. So in archaeology, we look at, we treat proteomics very differently to clinical practice, where in clinical proteomics, you're obviously able to look at over or under expression. For us, when we talk about amino acid composition, we're purely talking about the relative proportions that we've recovered in terms of the proteins that we're seeing. So we are seeing enrichment of leucine in ancient brains, in the sense that when we compare ancient brains with modern post-mortem tissue that's been digested in the same way in the lab, we see far more leucine in the ancient brains than we do in the modern brains, which is obviously we would expect to see some differences in ancient versus modern proteins, but there is no precedent that we've been able to identify in the archaeological literature for such a relative abundance of hydrophobic or branch chain amino acids like leucine and alanine.
Okay, let me think this in a different way, or formulate it in a different way. Shouldn't you really compare just the I call little bit of protein that are leftovers from a digested protein schnipsels, which is just German word for small pieces. Shouldn't you just look at the exactly same schnipsels to and then compare the amino acid composition? Because if you imagine that transmembrane proteins will be attacked on the extra membrane parts, lose those, so you enrich for the alpha helix, as you said, which are rich in hydrophobic and therefore branch chain amino acids. You will get an enrichment for these just because you only look at the fragments that were preserved within the membrane. So you should really, I think, only compare this composition to the same subset of protein digest schnipsels that you get from a modern brain.
Yes, that is what we're hoping to do in our project, obviously to make a meaningful comparison. I thought the fact that you mentioned intracellular and extracellular regions was a very pertinent point. Obviously, we do see compositional biases in the sense that residues, for example, more polar residues are heavily suppressed, whereas non-polar residues are more enriched in cytoplasmic versus extracellular locations, and that would certainly influence the amino acid composition that we end up seeing.
Yeah.
So yeah, you're absolutely right.
The other thing, I'm dominating here. I hope somebody else will jump in a minute. The other thing that I wonder, you have that down on your slide here, the citrullination of arginine. That's quite an uncommon sort of modification in mass spec. I don't think most people would actually not look for that. Do you think that is something that happened during the lifetime, or is that more likely something that could have happened during the decomposition of the body?
That is what that's what we're hoping to find out. Our expectation at the moment is that we think it is an in vivo post-translational modification, simply because we don't generally observe it in other archaeological substrates. We specifically looked because the difficulty of presenting this data, as I hope you'll appreciate, is that we don't understand the way in which the brain is preserving through such extraordinary lengths of time. So we're keeping an open mind. We don't know exactly what is happening post-translationally to the proteins that may be biological or diagenetic. But in the case of citrullination, because, as you say, it is an unusual modification, something we don't normally see in archaeological material, we tentatively suspect that it's an in vivo modification, such as we see in, for example, Alzheimer's or in rheumatoid arthritis and the like. But our first and foremost question that we need to answer is by what means is brain tissue preserving through time? Because if we can identify what's happening to the tissue post-mortem over millennia, then we can really begin to meaningfully interpret some of these changes.
So why are these people preserved?
I'm sorry.
Let me just ask this one question. So why are these people preserved? Have they been embalmed, or did they fall into some sort of pits, or what are these?
No, as I mentioned, the ancient brain tissue is extraordinarily common. So far, I've identified over 1,300 brains reported preserved in a variety of different archaeological contexts. So we find them in wet and dry environments, aerobic, anaerobic, acidic, alkaline. To date, we've looked at proteomically the brain of Ötzi the Iceman. Obviously, that's a well understood mechanism of preservation, freezing. We all do that with food in our refrigerators and what have you. We've also looked at a brain that's 2,500 years old from a waterlogged pit. We clearly wouldn't expect to find such a labile tissue preserved in such a strange context. And we don't understand the mechanism by which the tissue is preserving in that kind of environment. What we imagine is happening at the moment is the process that's actually has parallels both in petroleum geochemistry and also to an extent in neurodegeneration. So some kind of process of oxidative cross-linking between proteins and lipids. The brain obviously is an unusual organ, 80% water, the remaining 20%, roughly 50-50 proteins and lipids. And if those degraded labile biomolecules are in some way cross-linking and undergoing this cascade of polymerization reactions, perhaps similar to what we see in neurodegenerative diseases, then they might actually represent extreme end members of certain aging trajectories by which they're forming this highly hydrophobic, resilient tissue. But as I say, it's all to be discovered. So,
I have a question, if I may.
Yeah, Stefan, go ahead.
Okay, thank you. I was wondering how different is the yield from your mass spec from an old brain compared to a new brain? So how many proteins do you get?
We're getting around 800 at the moment, which is about a quarter of what we see in fresh post-mortem tissue. But 800 proteins is utterly unheard of for archaeology. The next most abundant substrate that has a published proteome to date is only around 100 proteins. So it does appear to be an extraordinarily rich source of biomolecules. But the real curiosity for us is, as I outlined in my slide, that we're getting higher sequence coverage of certain proteins in ancient brains versus modern, although, as I mentioned, we're getting fewer proteins overall, which does suggest there's some kind of a preferential preservation going on. But again, it comes back to this idea that we don't know the mechanism by which these proteins are being preserved in the first place, but it's a good starting point.
Thank you.
I have a few. I don't know.
I was going to ask, it's known in the mouse, at least, that saturated fat very rapidly induces hypothalamic inflammation. So I was wondering if you can detect some markers of inflammation, which could hint on the dietary lipid composition.
I would love to be able to answer that question. I'd hope to be able to. We're conducting lipidomic experiments at the moment, but our preliminary investigations suggest that the tissue is highly fatty. But again, we don't know whether protein lipid stereochrometry is preserved, for example, or whether the relative proportions of for example, the highly functionalized fatty acids we would expect to find in the brain, whether those ratios are preserved.
But I was more referring to proteins associated with inflammation, markers of inflammation, rather than the actual fatty acid composition of the brain.
We just don't know at the moment. We haven't been able to conduct the lipidomic experiments yet.
Okay,
we'd like to do.
I think I need to stop there because we need to, I'm conscious that time is running and we have 3 other speakers, so we need to share over. So it's thank you very much, Alexandra.
Thank you, I'll stop sharing.
Okay, and it's Anthony then, isn't it?
It's supposed to be. Yep, give me a sec. Let me see. I see it. Great, thanks. So good afternoon, everybody. My name is Anthony Zhang from the University of Cambridge, the Institute of Metabolic Science, Carbon Spray Lab. And so one slide summary. So as Carbon have given a talk this morning, one of the research interests in our lab is to look at the protein sensing mechanism in the brain. So in the past, we have identified that the NTS, the nucleus of solitary tract, the hindbrain, is an important region for protein sense to sense the dietary protein as well as mediate the anorexic response of the high protein meal. But at that point, we didn't really know how it works. So in the current work, so we followed that up. And we use a variety of genetic tools to identify there is a new hindbrain to forebrain neural circuitry that mediating such observation we observed in the past. And more specifically, it is the NTS-PLRP neuron, the prolactin releasing peptide connect to the DMH leptin receptor expressing neuron, which wire there, and it connects to the arcuate AGRP neurons. And this would mediate the high protein meal with the anorexic effect while suppressing the activity of the arcuate AGRP neurons to reduce food intake. So basically, that's what this story is about, and more details on my poster. So that's all.
Open. Thank you very much, Anthony. Open for discussion. Who wants to start us off? Right.
I can have I have a short question. So do you think, now you work with a branched chain amino acid, do you think that other essential but also non-essential amino acids can go in and affect food intake in similar ways through similar circuits?
There are different amino acids. So we have characterized, we and others have characterized a couple of them. But it is the leucine that has the most potent anorexic effect in the brain. Whenever you inject it from literature, you can find it consistently suppresses food intake. When you give it in the ventricle, or to give it in the hypothalamus, for example. Yes, different amino acids have different response, but how that actually relevant is another issue though.
But can it cross the blood-brain barrier, or do you have to inject it directly in the brain?
Can it cross the blood-brain barrier? I think it is yes for this one. But I don't think there's a very well described mechanism yet. Correct me, Cameron, if I see you are here.
Yes, it does cross the blood-brain barrier through the LAT1 transporter that is heavily expressed on blood vessels and in the brain.
Yeah. Anthony, if I may, how do you fit this in with Zach-Knight sort of stuff, that the AGRP neurons actually shut down just when they see the food and then come back on? Do you think this is a secondary sort of signal onto the...
I think it's an integrated signal. So in the Zach-Knight paper, they actually saw it is also via the environmental cue, which later on it has also been described. It can be via the DMH as well. So I think the DMH is one of the integration sites to regulate the AGRP neurons. So that's how we see.
If I may just add something here, Zach-Knight and others also found that direct infusion of nutrients into the gut suppress the activity of AGRP neurons. So these neurons are initially suppressed by food-related cues, and then ingested cues and circulating cues. So there is a different time frame for these different cues to suppress the activity of AGRP neurons.
Okay. Yeah, no, that's fine. I completely agree. I asked you an hypothetical question because I had seen this story before. On figure 3, which we can't see, that's a bit unfortunate. If you do the percentages, I got the impression that actually most of the cells that are FOS activated after your leucine injection, they are actually not the PRRP neurons, are they? They at 70 or so cells, if one goes through the numbers, are something else. Do you have any idea what they are?
We didn't. So we know it was about 30% of all activated neurons in the NTS. They are PRRP, around 35%, 1/3 of them. And for others, we don't know, but that would be very interesting to explore, particularly with the technique we use activity capturing. And that's the idea we use it to begin with, because we think it's more representative to what we are looking at. So
but you haven't yet looked, or you don't really want to reveal what the other neurons might be. I mean, there are loads of other neuron populations in the NTS that might be.
We know the TH is a big population. We know that they are, which PRRP is also a subpopulation of them. For others, we know POMC neurons. They also are 1/3 of all the POMC neurons. They are activated by leucine as well. For others, we don't know. We haven't done a very robust characterization yet so far. But the activity-dependent capturing would be a technique to go for if we really characterize the neurochemical identity.
Can I ask a quick question?
Yeah.
Do you know the mechanism by which leucine is actually regulating the neuronal activity in the direct?
So you mean the molecular mechanism? Is that what you mean?
Yeah.
So Cameron in the past, she has characterized the mTOR is one of the important pathway. I mean, as you read the literature, you will know leucine can directly activate mTOR. That's one of the important pathway. And the ERK pathway is also another, it's also an important one. So that is some of the pathway we know.
But like when does it diffuse through like the cell bot? Like how does the neuron recognize the leucine concentration? Like the ones in the NTS?
Oh, so the neuron, so the leucine can get into the cell, and in the cell it can activate the mTOR. That is one of the pathways we know, but we will expect more to discover.
Okay, cool.
What happens like if you have these like PRP neurons? What happens if you like inactivate them using, say, like optogenetics or chemogenetics? Has that been done?
In the past, well, I mean, we have in our pretty recent publication. So we not exactly PRRP, but that is the calcitonin receptor expressing neuron. We silence them. We use tetanus toxin. We silence this group of neurons. We know they heavily overlap with PRRP neuron in the NTS, and we also know they are responsive to leucine. When we silence it, the leucine and the resting effect is basically gone. So we think they are very important.
But does it like by itself already affect the food intake if you silence them?
It does. It has we have done it, but our collaborator they have characterized the calcitonin receptor expressing neuron in the NTS, and that they are more susceptible to high fat diet induced obesity.
Thanks.
Sorry, so you were saying that you think like the mechanism that leucine kind of just enters the cell and then regulates mTOR signaling, but why would it specifically target different populations within? Why do you think it would have specific effects on specific populations within the NTS?
Well, absolutely good question. I mean, that would be the same for the glucose as well, the glucose sensing. I mean, basically all the cells, they have the ability to sense glucose, but I mean, some cells are particularly responsive to glucose. And that is one of the mechanisms we want to explore, really. So,
okay, so you don't, still looking, cool.
Still looking, definitely. I mean, again, the activity dependent labeling thing is a very, very interesting idea to go for, to really look for the mechanism as well.
Cool.
Shall we move on to the next poster from the Blue Group? Thank you, Anthony.
Thank you.
And I think it's Danae.
Yeah, sorry, I was muted. Hello, everyone. Alors, here we go. So hi, everyone. I am Danae Nuzacci. I work as a postdoc in the Blue Lab. And today I am going to show you some of my previous work. So in that work, I explore what's happening around POMC neurons before and after a meal. And for that, I use different models of modelization of the prandial state in the mice. And I coupled all these models with parameters to evaluate the neuronal and glial plasticity around the POMC neurons, such as the neuronal activity, the synaptic coverage, and the astrocytic coverage of POMC neurons. And what we find is that compared to the preprandial state, after a balanced meal, you have an astrocytic retraction around the somas of POMC neurons. And this is provoked by the hyperglycemia in postprandial. Also, this is correlated to an increased activity of POMC neurons. And interestingly, it is also correlated to a modification of the feeding behavior, because when I increase artificially this retraction around POMC neurons, I have more important and more long feeding episodes in the mice. So that could suggest that this glial plasticity induced by a bolus of glucose in postprandial around the POMC neurons could be a daily mechanism to regulate feeding behavior and satiety. And that's it for me.
Okay, thank you very much. Open for discussion.
I guess I just have one quick question.
Yeah.
So, or just to start, so in your graph, or like the first figure you show, you indicate that you used a high sugar diet mouse or high sucrose diet mouse. Is that what the HSD?
No, It's
No,
a standard diet.
which diet is HSD?
HSD,
Standard.
I don't have a HSD diet. I don't think so. It's 1 hour standard diet or 1 hour high fat diet.
Okay. I just seen 4 groups in the postprandial group. Okay, and I just didn't see that one show up in the rest of the figure. Okay.
I have a question. Thank you for a nice presentation. I was wondering whether the total energy intake changes.
No, there is no change in global energy intake in the standard diet group or in the high fat diet group.
And there's no effect on body weight either, depending on
In
how
1
they
hour,
choose to
it's
eat.
very unlikely that there is an effect on body weight. So no effect.
Okay. Thank you.
You're welcome.
Dani, I have an understanding question. So the astrocytes retract.
Yeah.
And you get a reduced inhibitory tone onto the POMC neuron. Why is that?
An excitatory. We
Yeah.
increase the activity of.
You get less iPSCs, don't you? So you get a
Yeah.
reduced inhibitory tone after
Yeah.
the astrocytes retract. So I don't quite, I would have expected if astrocytes retract, you would have bigger or more possibly synapses onto the POMC neuron. So I don't understand why that should reduce the iPSCs.
In fact, I think it doesn't change the number of synapses or the synaptic coverage in this situation.
Mm-hmm.
It's not about like a decrease of inhibitory input from the astrocytes onto this neuron into this situation, if that makes sense.
Okay, I can just come up with something that maybe the astrocytes quench whatever comes out of the inhibitory synapses onto the POMC neurons, but because I'm not aware that you would get iPSCs from astrocytes onto neurons, but that might.
No.
Okay. Anyone else? I mean, this is very elegant work, very difficult, I guess, to proceed with this. A technical question, which is quite challenging. So when you use your electrophysiological recordings, that's done. You isolate the slices an hour after your intervention, correct?
Yeah.
Yeah. But then it will take at least an hour before you get your first recording. So why does the slice not actually adapt to the new sort of environment that it experienced during that time?
Yeah, so we considered that we all treated all the maintain alive brain slices the same way, that there is like no effect of this whole treatment to conduct to this recording. But yeah, it was definitely like a question I had when I designed this protocol. Yeah.
Okay, I mean, it's your observation. It's just surprising, but and should need to take. So if we start doing brain slices in the lab, as you know, we are planning to, we should actually take this into consideration that maybe more is preserved than one thinks after the intervention. Any other questions?
Yeah, Danae, I have a quick question.
Yeah.
So with your astrocyte retraction, do you think it's like, how is that mediated? Is that via the astrocyte sensing something, or is it as a result of the neuron activity changing?
I think it's more about nutrient sensing from the astrocyte itself. Because I did this experiment where I block the uptake of glucose in the periphery, and it totally blunts the astrocyte retraction. It blocked it. So we think that it's more about glucose sensing by the astrocyte than directly by a communication from the neuron to the astrocyte. But it's a very, maybe it works in the two senses. We don't know. Maybe the astrocytes and the neurons are sensing glucose at the same time and can exchange information to provoke this retraction, because there is no effect on the astrocytes around the AGRP neurons, for example, in the study. So it seems to be cell neuron specific.
We have a little bit more time. One more question. I don't want to go to our poster that early. Well, if not, then thank you very much, Dani.
You're welcome.
The last poster is from Emily, which is from my group.
All right, just sharing. Can you see that now? Oh no, all the text is gone. Okay, it's here on my screen. Let me just, sorry. I'll try one more thing, and if that doesn't work, I'll just make it up. So yeah, as Frank said, I'm a PhD student in his lab at the Institute of Metabolic Science in Cambridge. I'm just going to share it from the actual PowerPoint. I have no idea why it's not there. Is that better?
Yeah.
Can you see? I don't think I can make it any bigger than that, but hopefully that's okay. So yeah, I've been working currently on a gut hormone called Motilin, which plays a really important role in kind of stimulating gastrointestinal contractions and motility. And there's been some evidence that it might play a role in kind of hunger signaling and potentially glucose homeostasis and insulin secretion, but the evidence on that is very tenuous. Interestingly, Motilin isn't expressed at all in mice or rats, nor is its receptor. And there aren't any sort of cell lines that express Motilin that can be used. So there have been very few studies over the years characterizing the factors involved in Motilin secretion and also kind of the intracellular mechanisms of the Motilin secreting M cells. So to get around this, we've recently started using human organoids for the study of enteroendocrine cells. So I've using CRISPR generated a human organoid line, which is labeled in the Motilin cells with this fluorescent protein VNS. And that allows us to kind of do FACS experiments to purify these cells, and then we can do transcriptomic, potentially peptidomics, to see what these cells are expressing. We've also generated some functional models. So we've expressed the calcium sensor GCAMP in the Motilin cells. And we've also developed a new mass spectrometry method to allow us to measure actual Motilin secretion from cultures. So what we're now hoping to do is kind of combine all these new models and data sets that we've generated to kind of characterize the physiological factors which might regulate Motilin secretion, which hasn't really been done before. And the fact that we can use a human cell model is particularly interesting. Yeah, so it's kind of a new model to study the enteroendocrine system, and we're hoping to use it to uncover some new physiology. So if anyone's got any questions.
Thank you, Emily. Open for discussion.
So which data suggests, sorry, which
No,
data
go ahead.
suggests that Motilin may have orexigenic effects or incretin effects? In which model has this been?
So it's mainly been done in human clinical studies. There have been, so in terms of a small animal model, the kind of house shrew expresses Motilin, so it's not quite a rodent, but it basically looks like one. But most of the studies, there's a couple of groups working on it clinically. So in terms of the orexigenic effect, they've sort of measured the contractions throughout the GI tract in parallel with sort of an objective hunger score, and use that to correlate Motilin concentrations and increased hunger. And they've also injected erythromycin, which acts as an agonist at the Motilin receptor, and shown that that can sort of increase spontaneous meal requests. I think the insulin studies, there was one in dogs and one in humans, but that's like, there's only 1 or 2 studies out there.
Mm-hmm. And is it known to which receptor it may act?
Yeah, it's called the Motilin receptor.
And where is it expressed?
So in the gut, it's mainly in the smooth muscle and enteric neurons. There's some evidence from rabbits and a little bit from humans that it might be expressed in regions of the brain, potentially the cerebellum and maybe the hypothalamus, but there's not a lot of evidence on that. So that's something we'd like to look into a little bit more.
If I may, I think the idea is more that the motiline, the initiation of stomach movement gives you a stomach rumble, if one wants, and that makes you hungry indirectly. That's
yeah.
I think what they think.
So
Yes, but if those could,
or not at all?
sorry, can you repeat that?
Is it linked to ghrelin? Is it co-secreted or?
Yeah, it's the same cell.
It's expressed in the same cells, but it's not so much in the stomach where ghrelin is, and we're not sure if the intestinal ghrelin is as active. But with the hunger contractions in the stomach, if they're caused by motilin, they cause hunger. But if you get stomach contractions caused by ghrelin or inhibiting acetylcholinesterase, even though you get the rumbling stomach, you don't get the hunger. So it's not quite clear exactly how it's working. I think it's probably got a subtle role.
Mm-hmm.
I have a question. So my question was also regarding the receptor and where it was expressed. But a follow-up, is there any pathological diseases where it either increased or decreased? So like leptin is increasing obesity or something like that? Does the physiological response changes depending on disease state?
There's no sort of specific diseases associated with kind of defects in motilin signaling. There have been some studies looking at kind of people with obesity or type 2 diabetes and saying that it might be slightly higher. But yeah, I don't know of a clear link with any particular pathology. It's one thing which, so for various hypomotility disorders, motilin's been looked at as an agonist. So things like diabetic gastroparesis, and potentially for kind of critically ill patients who have trouble like clearing out their intestine, it might be that motilin agonists could be useful there.
What about something like gastric bypass? What happens there?
So there was one study which showed that it went up kind of immediately after, but then by a year later, it was kind of back down to normal levels. Obviously, Roux-en-Y gastric bypass, the kind of most effective form, bypasses most of the duodenum, which is where most of the motilin cells are expressed. Our lab has done a couple of studies looking at this, particularly in lean patients, and there's not a massive difference one way or other. It might go down a little bit.
I think we should also point out that the existing immunoassay that you can get for most of them are not very reliable. So that's why we have developed this mass spec technique. So I wouldn't read too much in what people measure with motilin in after gastric bypass, because mostly it's not measured because there isn't a good assay.
So it's Irison story, kind of.
What, sorry?
It's just the same thing with Irison, another hormone where
yes.
Right.
Yeah. A lot of assays that doesn't work.
Yeah, so we're hoping that our new mass spec method will help us to kind of be able to properly quantify it.
Great, thank you.
So I got a question. So how is motilin secretion regulated? Is that you have done some phosphocholine treatment thing? So is
yes,
it
so
regulated? That's the question.
I've just done a couple of pilot experiments so far. That's going to be the rest of my PhD, is trying to figure out how it's regulated. Yeah, all the evidence currently is based off kind of early clinical studies in the 70s and 80s, where they kind of gave humans different meals and saw what happened to their plasma motilin. Again, as Frank said, measured with fairly unreliable assays.
Mouse, I mean, there must be some fast refeed mouse tissue around in the lab, in the gut, and just wonder whether it is sufficiently regulated in this sense, the motilin.
Sorry, did you say mouse?
Yeah, in mouse.
So in mice don't express motilin at all.
Oh, okay, I see.
So I think, yeah, they only express ghrelin, which might take over some of the roles, which is why it's not really been studied very much today. And a lot of the existing data sets, particularly for like single cell analysis of the brain, obviously don't have.
Are they similar structurally or signaling pathway? Are they similar?
They're similar, and they're kind of evolutionarily linked, but they don't cross-react to each other's receptors.
I see.
That we should, Emily, you should probably say that motilin also is regularly secreted, right? So it's not just so not just postprandial regulated, but it plays an important role in the migrating motor complex along the gut. So when you don't eat, your gut squeezes every hour or so, and this is initiated in the stomach or in the duodenum and squeezes all the stuff that has accumulated from dying cells down towards the exit. And motilin plays an important role in that initiation of this migrating motor complex. And then therefore it needs to be secreted from some sort of inherent sort of mechanism. But then it gets inhibited, is my understanding, after a meal. Is that right?
It depends on the composition of the meal. So
Okay,
people have shown if it's like a fatty meal, then it will stimulate motilin, whereas if it's high in glucose, it'll inhibit it. So I think it's kind of two interplaying regulations. So there's the kind of regular cyclical MMC, and then that gets interrupted. So that normally happens kind of every 90 minutes. It squeezes everything down, but then that gets interrupted every time you eat. So then the postprandial response might differ depending on the composition of what you've eaten. And interestingly, water alone can stimulate motilin secretion. So it might just be stretching of the duodenum or osmolality changes or something. So I think we have to take some of the studies with a pinch of salt.
And what's sort of the half-life of motilin? How long does it hang around?
4 minutes.
4 minutes, okay.
It's not very long. But that's similar to most other gut peptides, or a lot of them. So.
Well, I have a general question. So, I mean, given mouse can't express motilin, so, I mean, what would be the in vivo model you would use? I mean, you can't do human, right, I suppose?
Yeah, so, well, you can do some things in humans. People have done a lot of stuff in pigs and dogs. I don't see us getting pigs in Cambridge. But, yeah. I mean, given that we can measure it in human plasma, like it's very feasible to measure the motilin response to a meal or whatever, a patient that's been given an OGTT. But obviously more manipulative things we can't do in a human, which is, where you'd have to go to either the shrew or the pig or the dog. But yeah, that's
why very interested in that shrew, aren't they? I mean, I'm going to start with that, but that I think that is the same shrew that also does the vomiting, isn't it?
Yeah.
So I don't know if you guys have seen that. This has been neurobiology things in the recent years. It looks like a mouse has a bit longer nose, but it can actually vomit, and they like to show that video where it actually does that. So. And from that point of view, it's a good model that people now are very interested in spread in pharma. So.
We have to find it in the foothills of Nepal, I think. It's not really bred in labs. But that's, I mean, why it's really good that we've got this human cellular model, because then hopefully anything that's species specific we'll be able to pick up in an in vitro model.
Okay.
Yeah, I think that's it.
Okay. Thank you to all four of you. If anyone has any more overarching questions, they are welcome to voice them now. Otherwise, I think we are ready to leave and join the closing ceremony for today. Okay. So I see you all then outside the room again.